process, which is muscularization of the diaphragm. Because if you don't have proper muscularization, you'll have eventration defects. So you'll have a very, very thin diaphragm, and then the abdominal contents can herniate into the chest cavity. So there's different aspects of the developmental process that will lead to different kinds of diaphragm defects, different locations, and we're still trying to sort all this out because it's not known. It's very unclear. And even muscularization, we know certain things happen, and we also learn from the limb in that aspect because the limb bud is muscularized in a similar way in the beginning. So we learn from the limb, but it's one of those where we're sort of, we know a few things, but we don't know a huge amount. So this is just showing you one example, and it's muscularization at 11.5. So at the same time point that we've been talking about so far, I don't even know if you guys can even see this at all. It's really white. But you can, what I did was use a special antibody for PAX3, which is a muscle precursor cell marker. So these are going to be muscle cells eventually, but it marks these precursor cells that are destined to go to the, that are going to go to the diaphragm. So they follow a special path. They go to the limb, and they also go to this pleuroperitoneal fold region. So if I make it bigger, hopefully this region, you can see all the dots in brown? Those are muscle precursor cells. That's PAX3 expressed by these muscle precursor cells. So the muscle cells are going and migrating on top of the pleuroperitoneal folds, expanding, and they're going into their correct orientation, and there's guides, there's signaling cells that are telling them where to go, so it's a huge process. And we don't really understand the whole process, but we're bit by bit. So this is at E11.5, so then a day later, this one is kind of hard to see, but one day later in the mouse at E12.5, these are the regions of the developing diaphragm now. And you can see the dots here, and you can also see dots of muscle cells forming around here. And we're using a marker called MyoD, which shows muscle precursor cells have now entered into the muscle pathway. So they know they're going to be muscle cells. So they express a special protein that says, I'm going to be a muscle cell. So it's going to enter into this pathway. So we can follow development of the muscles in the diaphragm. So that's what I'm just trying to show you, is following the developmental pathway. So then one more day, and then I won't show you. Now, at E13.5, we have nice muscle cells. And this is taken at, you can't really see it, but the embryo is sectioned. Now they're larger embryos. Now all of these are under the microscope.
Each of these has to be a different mouse embryo, or are you following the same embryo?
You can do co-staining. You can stain with different.
No, it has to be. It has to be means like once you section it, it doesn't grow anymore because it's in the box.
Yeah.
Yes. Oh yeah.
You sacrifice
yeah.
it.
So it's
Okay.
a very, very time-consuming process. It's very tedious, and they're actually like microns thick are these sections that we make. So we use teeny. And they're little, and then we put them onto microscope slides, and then we use the microscope, and then we take pictures from the microscope because you can't even see these. So I'm really, they're like your fingernail.
And do you make the embryos in a dish, or do you harvest them from?
We harvest them.
Good Lord.
But
Well, they're special lab. I mean, there's,
Yeah.
it's
the thing
not.
with those mice is that they have been, those are called strains, they've been in the lab so long and mated them with themselves so long that now they're genetically homogeneous. So it's not like they're very different from one another. You look at different embryos, but they basically have 99%
They're
the
the
same
same genetic
DNA.
background. They're the
Yeah.
same.
I was just trying to get, because it just seems incredibly tedious. So
It
I mean,
is-
and I
it's
don't mean to
enormously
belittle it.
amount
It's
of-
just
it's-
wow.
It's been a lot of work. So
I have a question.
Yeah.
The 11.5 equals week 5?
No, 11.5 is 11.5 days.
Yeah, I know,
after
but
the
in
human.
mice.
In the human,
But
in the
in the
mouse, it's
human.
about 5 weeks, 4 or 5. I mean, it's not exact, but it's about probably 5 weeks.
Because I was wondering, in my case, I started bleeding in week 5 and I stopped in week 10. So,
This is a process that goes on within the embryo. So probably the bleeding was outside in the placenta or who knows.
I don't know, but it's funny. It matches the days or the weeks that where the diaphragm is forming. So maybe it's a coincidence. I don't know.
It's hard to
say.
tell. Yeah,
No, I
exactly.
know it's hard, but I was wondering.
But it's
you know?
interesting, yeah, the timing of, yeah.
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 It's interesting.
So this is just showing you Desmin is another antibody. So we use many different antibodies for many different things, and that's the reason why we use mice, is because we have an easier time with getting antibodies and stuff instead of using rats. And that's just, and rats are bigger, and it would be way easier to use rats, but they scare me. And can't do that. I'm afraid enough of mice. So you can see here, this is the diaphragm, and you can see the muscle cells, and actually Desmin is involved in the architecture of the muscle. So it's pretty nice. And the staining is red this time because I use a different kind of stain. Okay, so we use this, I just talked about normal development. So I'm not really going to cover abnormal, I'm not going to cover mutant models of mice this time, but we can use the mice then to go back, and let's say we find candidate genes that we're interested in, or genes that we think are important for, perhaps it might be causative for CDH or something else. We can create a mouse where we knock out the gene, where we get rid of its function in that particular mouse strain, and then we look at the mouse embryos again, and we say, oh, we can look at the whole embryo and say, oh, is it the diaphragm, is there a diaphragm defect, or are certain things not being expressed appropriately? Do we have weird patterning all of a sudden because we knocked this gene out? Maybe the muscularization isn't working appropriately, or it's not forming, or you have other issues of, all different kinds of other developmental programs that are going on. So that's why you can use your mouse, and you can say in your human population and your mouse population, you go back and forth and sort of learn from different areas. So now I'm going to talk about how we find candidate genes from our bioinformatics work and our computational work, which is because we do all sorts of studies on each patient, and then we have tons and tons of data, millions of data points in the end, and you have enormous amounts, and then you have to sort of say, okay, how do you pick and choose? Where do you go from there? And that's why it's been a challenge with all this data that accumulates, sorting through it, because you can do all these really cool techniques, and then you have to actually figure out what you have. And so bioinformatics have been helpful because they're a way to understand biological processes. And then when we do computational biology algorithms, we actually are able to develop new algorithms and mathematical formulas. And we use statistics to assess relationships between different types of large data sets. So when we do CGH analysis, we get one type of data. When we do SNP arrays, we get others. When we do our sequencing, so it's a way to combine all the different types of the platforms we use and techniques within our CDH cohort to try to filter out what's really going on and to find candidate genes. So this is a hypothesis generator. It's not necessarily giving us the answer. It's giving us areas to look into for developmental pathways and to try to figure out where we're going with studying particular genes. Because each gene you choose to study is a commitment. So you have to say, okay, this is very interesting, this gene, but you put a lot of time and effort to make the mouse model. It takes a year. So, and a lot of money. So you really have to pick and choose how you really want to study, how you want to follow through with your candidates, and which ones you want to follow through with. So what we use are one of the algorithms that is created in our laboratory. We have a postdoc who's a computational biologist from the Technical University of Denmark, who is working on congenital anomalies, and he's doing CDH, and it's great because he's taking all of our data and making all these different algorithms, and he has designed a protein-protein interaction network that he calls PPI. And the protein-protein interaction network can kind of be compared to, we can say it's a pretty simple concept, and it's compared to the idea of a social interaction network. So if you think about different social groups, actors might hang around actors more commonly than you would say, and then you have like politicians that might hang out, people doing different sports like golf, or, but typically you would say that Brad Pitt might not hang out with Wally over there. He probably would be the same social circle. Or, and actually I don't have Angelina